Okay.
Okay. Good afternoon, good evening, or good morning, depending where you are now. We are starting our part of the plenary session. And at the beginning, I would like to remind the schedule. Each speaker has 20 minutes for his presentation, and then 3, 4, maximum 5 minutes for questions and answers. I'd like also to apologize in advance if I will pronounce names of some speakers incorrectly. The only excuse which I have that my own name is pronounced in different countries differently and usually incorrect. The first speaker is Maxim Pogorelov, from Sumy State University, city of Sumy in Ukraine. He also represents here the University of Latvia in Riga. And the title of his presentation is Tissue Engineering Scaffold Development, Cooperation with Europe. Welcome, Professor Pogorelov.
Thank you, Anatoly. Thank you, Igor, for inviting me to your event. In my opinion, you're organizing the best networking event in Ukraine. You can bring together absolutely different people from absolutely different fields, and it makes us, all of us, more powerful, more stronger. When you invited me to this conference, so I'm thinking what I can share about the sustainable development. I decided that cooperation, okay, I feel like this, I trust this, that cooperation is the main factor of sustainable development, the main factor of sustainability. And in this short presentation, I would like to demonstrate how the cooperation with Europe can support research in Ukraine, or whatever, not in Ukraine, in any country. The research stays on cooperation. Cooperation is like a main factor of sustainability in research. I would like to show, start from the scratch. What is tissue engineering? Because I suppose that not everybody know what is it. Not everybody from medicine or from biology field. So if you're looking for tissue engineering, okay, tissue and engineering, it's just a simple, we put together these two words and we try to engineer to build a new tissues. And the process how to build new tissue looks very simple. Every single tissue, if you look into our body, consists of extracellular matrix like scaffold and cells that grow inside. Okay, vessels, nerves, but anyway. To make the tissue engineering construction, the first step, we need to isolate the cells from the patient. It's like an ideal case. To seed and let cell grows. Then put the cells on appropriate scaffold, put to bioreactor or to incubator to develop this tissue, and then the last step is to implant this tissue to patient. In my opinion, both cells and scaffolds are important for this purpose, but scaffold is like a key factor of successful tissue engineering. It should be biocompatible, enough porous, with appropriate fiber size, with appropriate degradation time. In ideal case, it should be antibacterial, and for some tissues like heart nerves, it should be electroconductive. To make tissue engineering scaffold, we try to mimic evolution. You can imagine the billions of years of evolution when our body starts to grow from first single cells, that rise billion years ago. Then cells assemble together to make like multicellular organisms, and some fibers arise around the cells, and step by step they build human. The question is it simple? Is it possible to make tissue engineering scaffold to do successful research in one lab? And the answer, in my opinion, no, it's impossible. Even in the best lab in the world, it's most impossible because it requires so different expertise. And I tried to make a simple list of expertise required to make successful tissue engineering scaffold. And you see that when it's scientists from nanoscience, biology, immunology, chemistry, engineering, medicine, cell biology, material science, and many, many, many others. And to put such people together with very strong expertise in one place, it's most impossible. It first factor, and second factor is that if we just boil it in one dish, in one environment, it's not a good development. So we need to bring some idea from outside. We need to share our idea with somebody outside and give insights from other people. And the best way for successful research is cooperation. In Sumy State University, we have biomedical research centers that we developed like 5 years ago. That already include cell culture lab, back lab, animal facility, fluorescent microscopy, ELISA, chemical, atomic force microscopy, electrospinning laboratory. We have access to some engineering solution neighbors laboratories. But anyway, it's not enough. We have, I can say that we have the best people in this laboratory. So I'm proud to my people. But again, it's not enough to be successful. And we have a lot of partners. Here, just a very short list from our partners that we collaborate, including tissue engineering as well. I'm happy that we're doing this not just in bilateral collaboration, but under different projects. There are we secured 3 Horizon Europe projects with very wide network. Okay, we have no partners from Japan right now and from Australia. All other continents, in our pocket. No, I mean we can collaborate. We can ask them. We have a lot of some bilateral projects. Erasmus, Jean Monnet project. We have, for example, recently last month, thanks to Sergei Kirilenko, we got 3 projects from NSF, from European Office of Aerospace Research and Development, from Eurozone. We try to use this partnership effectively to make good research. And today I would like to briefly demonstrate some of our recent results in tissue engineering. Well, maybe, okay, if you ask me, can I build some organ or some tissue, I can answer no, not yet. Okay, but I will show you some successful stories of our research. Last year, we published our first clinical results on application of CAD/CAM technology for building personalized scaffold that our partners successfully implement in several patients, like patient with Goldenhar syndrome, you can see here, and patient after the high energy trauma, a 5-round injury from a Kalashnikov gun. But to make this before we realize this clinical trial, we did a lot of research in more simpler scale. And for example, we tried to use metal scaffold with different architecture, because okay, if we would like to replace the bone, we try to mimic the structure of the bone. And no, unfortunately, metal now is the most good or suitable candidate for this scaffold with enough mechanical properties. But nobody knows how the architecture of the scaffold influence to bone regeneration. Architecture of metal also affect the mechanical properties, as you see, or destruction behavior of these porous scaffolds. But metal is metal. but we would like to make something more biocompatible, more similar to MON. And we applied methods that use called plasma electrolytic oxidation that allow to grow the ceramic layer over the metal. And we add calcium phosphate solution, add scaffold, apply high voltage, and we can see the formation of ceramic layer with such porous structure. And when I demonstrate this picture to some biologist and ask, what is it? Somebody even answer me that, oh, it looks like a bone. But no, it's not a bone. It's a ceramic layer that we made over our scaffold. Well, but it was not enough for us. We also added silver nanoparticle to this solution to make scaffold antibacterial. Not only good enough for cell attachment and proliferation, but also to give some antibacterial properties in case of infection. So, as you see from this slide, this is like balls. It's remelted metals after the building of this 3D scaffold, metal scaffolds. Also you can see, for example, on the right side, you can see the small light dots that are silver. And as you see, also the surface of this material is covered with ceramic layers that is made of calcium and phosphorus, as noted at these tables. And also you can see that some samples has silver on the top. And what is the most important, that we were able to make this ceramic layer inside the ports, not only outside the scaffold. Later on, we see the cells on the scaffold and look how the cells growing on and penetrate inside. And yeah, we have quite successful results, but unfortunately, silver nanoparticles not only inhibit the bacterial growth, but also affect cell growth. But in some cases, we can equalize the concentration of silver and can equalize the antibacterial action and biocompatibility. And using this data, we were able to make the scaffolds that we applied in real patients with really successful results. All patients after the operation for sure survive, and I hope now they are happy because they have new faces. It was first case. The next case of our successful story is cardiac tissue engineering. No, this story is more complicated. As you see, amount and structure of transplants performed in Ukraine in 2022. It just year after our government finally approves the law about the transplantation. But the number of transplantation, around 400, is nothing, because right now we have data that more than 5,000 Ukrainian need heart transplantation. But trust me, 5,000, it's not like real count, because I cannot predict the real number of needs, but it's much more higher. The statistic, for example, from US, that every 9 minutes, one patient added to the transplant waiting list. Every 9 minutes, one patient added. And to develop scaffold, we're looking for materials that are electroconductive, materials that cause MAXINs. It's another successful story of cooperation. Professor Yuri Gagosy, who is originally from Ukrainian origin, now he's director of Nanomaterials Institute in Drexel University. In 2011, he discovered new class of nanomaterials, 2D nanomaterials that calls MAXINs. And our story, our journey to MAXINs was quite long. The first time we met Yuri was 23 17th, it was in Zatoka near the Odessa during the conference Nanomaterials Application and Properties. And also there we met with Dr. Alexei Gagotsi, who is founder and owner of Material Research Center, the biggest producer of vaccines. And later on, we start to cooperate between Drexel and MRC, but later on, we start to cooperate from Latvia, Poland, Germany. We assembled big consortium, and currently we secure two big European projects, MXMAP and Esculap, dedicated for vaccines for biomedicines. Currently, there are a lot of publications about the application of vaccines in biomedicine, but one of our fields that we are more or less successful is scaffold vaccines loaded scaffold. Our idea was to load the porous electrospoon membrane with vaccines to make them conductive, biocompatible and conductive, and to use these materials as a scaffold for heart regeneration after the myocardial infarction or other cases. We built electrospoon scaffold using polycaprolactone. But the problem, you can see, the scaffold electrospinning is a unique technology that makes that allow us to make randomly oriented fibers like extracellular matrix. But the problem is that materials are quite hydrophobic, and to use in several technology, to analyze in several technology, we selected deep coating for vaccine immobilization. We use several methods to make the scaffold more hydrophilic, and after many attempts, we realized that oxygen plasma treatment is the more suitable and more good method to make this vaccine containing scaffolds. So we used different methods to analyze the structure of this material, the presence of titanium, because we used titanium carbide vaccines for immobilization. To analyze how they change porosity, we use scanning electron microscopy, EDS mapping. We cooperate in this regard. We cooperate with many institutions in Ukraine, in Poland, in Latvia, in Slovak Republic. For example, yes, this data with high resolution TEM was made in Slovak Republic, where we can visualize the single flake of vaccines. So we deeply analyze every single piece of this material to prove that structure is quite good for tissue engineering. We made my PhD student from Poland. She made a very good 3D reconstruction of this material and visualize. You can see it looks like real material. We analyze contact angles, hydrophobic or hydrophilic properties of these materials, and prove that addition of vaccines can decrease contact angle and makes scaffold more suitable for cell adhesion. The main question was, is this material electroconductive? Because do you remember initially our idea was to make electroconductive scaffold. So in Serbia we made several experiments, and we found that the conductivity of our materials is from 5 to more than 300 millisiemens per meter. That is quite enough compared to electrical conductivity of sinoatrial node or even Purkinje fibers. So another very important question is biocompatibility. Is the scaffold can support cell growth and proliferation? It was a long story, starting in Sumy, for example. You can see the first slide, it's just simple DAPI staining, and then continue in the University of Latvia together with Professor Una Riekstina. And we have enough information that proves that our scaffold biocompatibles, electroconductive, and support cell growth and proliferation. You can see the cells made just like simplest on the surface. So we try to find some antibacterial property, but unfortunately, no any antibacterial properties in our materials. And we can conclude that we successfully made the first step in development of electroconductive scaffold for cardiac regeneration. And I would like at the end, I would like to acknowledge all my partners and all projects that made our story successful. Yeah, cool. I'm in time, and there will be no any big conclusion. Just one conclusion, that cooperate, and together we stand. Together we can, with cooperation, we can do a lot of very nice things. So whatever we are, in Ukraine, in EU, in I don't know, in North Pole. So, and cooperation makes our life and our research sustainable. Thank you.
Thank you very much. Please ask your questions.
If I may, Professor, about Maxine, I know you are a huge proponent, yeah? As I understand, it's actually a form of lace, kind of form of nanoparticles, yeah? Am I correct?
Yes, Maxines, it's like 2D, it's a class of 2D materials. It means that one dimension of these materials is around in nanometer scale. But it has quite high lateral size, up to a few microns. It could be from 200, 100 nanometers to 5 microns in lateral size.
Okay, yeah, I got the idea. And do we have any risks from this scaffold? We will get these flakes around the tissues and maybe some negative effects.
Very important question and very interesting question, and we have an answer, unfortunately. As I told, the materials was discovered just 12 years ago, and biological investigations start in the first publication about the biological, the application of Maxine in biology and medicine was dated to 2017 or 2015. Currently, including our group, we try to understand what happens with Maxines after the injection or insertion somehow to insert in our body. We can hypothesize that Maxines, because Maxines oxidized and transformed from, for example, titanium carbide to titanium oxide. And theoretically, it will break up to the titanium oxide and remove from our body. But we need to prove this. Nobody knows.
Okay. And one more, if I wish, one more question. Do you have, I mean, this animal testing in first this scaffold examples, yeah, for bones? How long actually you have positive results? I mean, it years or something, or maybe in people, in patients.
Yeah, I understand. In this case, we didn't use animal. Why? Because we use materials that are already used in medicine, titanium. It was titanium aluminum vanadium alloys that probably some of you have in your teeth as an implant. And we not require animal testing. We prove biocompatibility. And we were able, after the ethical permit, we were able to use in human. But other materials, we had experience with materials for bone regeneration. Usually we're looking for up to 2 or 3 months. It's enough for clinical response in animal.
Okay. Thank you.
I have a question. What is the price? What is the price of the material?
You mean Maxines?
Hmm?
I don't know. Friendly speaker, no. Because we get these materials from our partner and we didn't pay. But you can check the web page of Materials Research Center and can directly ask them. But it's not like very huge price, but okay, still not cheap. Anyway. We have no mass production of vaccines right now. And you know, no, I cannot comment this question because we get from the partners for free.
Okay. I have one more question. Obviously, these materials are needed everywhere, but especially in Ukrainian current situation with the war, many people injured. What is the regulatory path to get into clinical use of such materials in Ukraine?
Frankly speaking, very hard. Very hard, and first answer and second answer is require some financial support. We cooperate with several companies in Ukraine and in Poland who okay, I can say that we developed these materials together with these companies. So it's not our intellectual properties, complete intellectual properties. We work together with companies, we shared intellectual properties, and the next stage company tried to apply for some regulatory agencies. And for example, the dental implant is already used, already certified in Poland. Dental implant with this plasma electrolytic oxidation coating. But this personalized implant, I think for personalized implant, the situation is more easy because you not need to apply for regulatory. It's like individual case with every single patient. When we use materials that already on market, like titanium materials, we can use for patient directly. But also, I'm not enough qualified to provide full answer to this question. Yeah.
Okay, thank you very much.
Thank you.
Let's go to the next presentation.
Okay.
Our next speaker is Dr. Andrew Civic. Is it correct?
Yeah, correct.
Good. Very good. From the National Center for Therapeutics Manufacturing, Texas A&M University from US. And the title of his presentation is From Theory to Practice Inside the National Center for Therapeutics Manufacturing. Welcome.
Okay. My name is Andriy, and I'm in Texas, and I will try to present. I'm not sure how fit this presentation is for our purposes, but I will try to make it fit. So I will share my screen, I guess, and let's go from there. So here we go. Not sure how to make a big screen. Okay. So National Center for Therapeutic Manufacturing is a unique organization, ladies and gentlemen, and I'm really pleased that I have an opportunity to present it here, and I hope that one day maybe some of you will be able to cooperate with us. So this institution was started in 2012, a long time ago. And it was designed for rapid response to biological threats as a part of governmental thing. And whole building is this big, huge thing. Now, right now, it's split in two parts in CTM, National Center, and Fuji Film Dies and Biotechnology. I'm sure you remember that company, Fujifilm, for making films. Right now, they are big international, I would say. They are located in Europe, in Canada, in America, in Japan. International biotech company. They're huge, and they conduct development and research for other people. They don't have their own product, but they manufacture things. And as part of our function, we were contract training for industry professional, for university and college students, and for rehabilitation of veterans, especially veterans, because those people from military, I served in the military myself, so I know that they are disciplined and they can follow the protocols and procedures, which is really crucial in biotech industry. Now, right now, we have like open enrollment, and we have contract research and development, and we have several different interesting projects, and that's why I was asking about the price of that scaffold, because one of the projects is related to that. So I'm not sure if I will be able to show you the drone tour, but I will try. Okay, this is the drone tour of the facility. Okay, I hope now you have some idea about our facilities, and I will tell you what we do. We are not teaching, we are not manufacturing, we are not doing research. We all of the above, and we are positioned quite in the middle in between all of this. So if people would require a research, they would come to us. If people would require teaching, they would come to us. If people would require some like pilot scale manufacturing, they would come to us. And here is our educational program from our school students to college students to professionals in biomanufacturing. We have many different courses, and those courses are available on demand. And we have some unique courses that I will be mentioning here. And especially it became evident whenever we started, whenever we went into a COVID situation. Fujifilm, that company was commissioned with production of vaccine, and they needed to develop a workforce very quickly. So we developed for them so-called boot camp, biopharma boot camp. In 5 days, we would train personnel just to understand what they have to do in this in the real manufacturing setting. So whenever you look at that tour and the presentation, we have upstream, downstream, analytical, and we have manufacturing suite and everything. So we were able, during very short period of time, we were able to train like more than 350 individuals that they will be ready for process. And it saved money for Fujifilm tremendously, because training on site is highly expensive and time consuming. We provided the program that was suitable for the company, and we were able to deliver that. So here you have all different types of courses. We have online format, we have different levels, and whenever you look at this introduction to pharmaceutical manufacturing, CGMP procedure and documentation, all different kinds of things. We have advanced courses for downstream, for upstream, and very recently we launched a unique course, which is mRNA vaccine manufacturing. And with mRNA vaccine manufacturing, this is the first hands-on mRNA training course in the United States. And we do teach how to conduct IVT reaction through cold chain. And we have unique equipment. When you look at this side, it's a robotic system, quantum, that is capable of generating from 1 to up to 5 grams of mRNA ready to use in 1 day. It's a huge capacities. And whenever you looked at our facilities, we have all the equipment necessary for the upstream, all different kinds of bioreactors, from wave bioreactor to small scale to large scale, from liters to 100 liters bioreaction. We have bioreactors, we have analyzers, all different kinds of analyzers for media analyzing, for cell viability, density, and the size, and all these things. We have very good microscopes. Here you have downstream equipment, and with downstream equipment, we have different types of chromatography systems, from small scale to like ACTA ready. This is a production system, production scale. We have all different filtration and tangential flow filtration systems. So everything is in place, and with analysis of whatever we have all different equipment to analyze the final product. And we, how do you call it, expanding the list of equipment that necessary, and right now we are in purchasing of mass spectrometer. Our staff at NCDM available for collaboration, for process development, and we do participate in many different kind of biotech startups. And we can produce in small scale, not large scale, but small scale, whatever you want to produce, starting from small molecules in bacteria, ending with, I don't know, recently we scaled up mesenchymal stem cell production for human transplant. We can develop all different kinds of assays and stuff like that. By looking at the program of the conference, I decided to include one specific project, very funny one to begin with, and it's cultured meat. So you can call it synthetic meat, healthy meat, cell-based meat, clean meat, artificial meat, whatever you call it. So it's the project that we are trying to grow cells and convert them into meat cells. At the beginning, I was very skeptical myself. How is it possible at all? And apparently, we have more than 30 startup companies that are very interested in that. And in our university, we have a lab that does this. And now we are helping them in cell line development and in scaling up of their process. So the process starts with embryonic stem cells. You can derive those cells from any type of animal, including fish and, I don't know, turkey or chicken. And then from that cell, you derive stem cells. Stem cells you can convert into any type of cells, and then you bank those cells. You create master cell bank, that's what we do. And then from master cell bank, you create working cell bank. And once you have those cells, you can differentiate them into myocytes or fibroblasts. And here you have a production train. It takes one while for the consistency, and then you have production train, and you produce ever larger quantity of the cells with whatever media is required, with all appropriate conditions. And they are multiplying very quickly. And then the last portion of this process is differentiation and formation of the actual structure of that meat. That's why I was asking about the price of the scaffold, because those cells can be deposited on the scaffold by 3D printing or just by differential plating on that scaffold, and then you can have that piece of meat. And this product is already used was generated in space station. So they are looking not only just for fun, because right now it's quite expensive. In order for this product to be suitable for mass production, the media should be fairly cheap. Like 1 liter of media should be $1. That's expectation. Right now, it's not the case, but they are working on that. And the composition of the media, the way you differentiate the cells, and the way you combine those cells, are still under research, because you can regulate media composition, you can regulate type of cells you're depositing, then you can regulate the outcome of your final product, and that product may be more nutritious. I'm not sure if it will be more delicious, but more nutritious, you can definitely make that product. So this is a short presentation. I don't want to spend much time, but I am willing to answer all the questions you might have for the institution like in CTM, because we in the middle, in between real researchers and between the big pharma. So we are trying to scale up whatever on the bench, and sometimes it's challenging because people, whatever they develop on the bench, they don't understand that the same process can be scaled to manufacture. So if you have questions about this, please ask.
I have a question. Thank you for amazing presentation. Just a few questions. First, did you test this meat?
No, it would be too expensive for me to taste it. Yes, right now the price is yeah. And the quantity is fairly small. However, why they are doing this? Because of environment, because of gas, like this green gas production. Yes, It still requires CO2 because it's mammalian cells, but it's like 10 times less of CO2 production, and you can regulate everything. And more than that, you cannot take cow to space, but you can take cells, and you can take like bulk of very solid like media. So right now we have solid media. You can have powder you use to suspend in water, and that's it. So, no, I haven't tasted that. I would like to, but
no, it's interesting experience, I think. Another question: do you have any state support, or just working with companies and grant center?
excellent question. So tomorrow, actually, we will have a representative from the government, and that representative will be walking around. Right now, we don't have a state support, but if you know BTEC in North Carolina, they do have a state support, and they like similar kind of facilities. They train, they teach, and they do small production. And yes, we are looking into that. And right now we are run solely on whatever we cooperate with companies.
Okay, great. Thank you.
Any other questions?
Maybe Pan Andrew, to continue this last question, your typical form of collaboration with research teams, yeah? It's kind of general, maybe, I don't know, mutual projects or something like that, or researchers try to find some financial support.
So, for example, I will give you a couple of examples. Recently, we had like one research lab and one startup company. So they came to us and say, do you have capabilities of generating cell lines? If you do have, then we include you in our research, or company will pay us to develop cell line. So the researchers, the university researchers, they have money dedicated to such process, and we producing again nanomaterials like fibers in bacteria, and they have money allocated specifically for scaling up projects.
So you are actually, am I correct that you are actually in, let's say, in searching of this team interested, I mean, researchers interested in collaboration or what?
Absolutely. So there is no point if you do any kind of research, you have to envision a commercial application of that. So if you're manufacturing a drug, you want to scale up. If you're manufacturing antibody or vaccine or cells, we were working with mesenchymal stem cells, you would like to make it commercially available. So they come into us and they say, is it possible to make it, to commercialize it, to make it a large scale? And before they make large scale, they have to produce like pilot scale. If it's feasible, if it's doable, then we can do it. Yes, so they are coming to us specifically. Is it scalable? For example, we had a project whenever they resuspend cells, small amount of cells, like 1 milliliter at bench scale, they resuspend in pure water. You cannot do that because cells are lysed. But at small scale, it happens, and the resuspension is quick. They exchange the media and everything. At large scale, we failed badly, so we developed a completely different process for changing media in between the processes. So yes, researchers are coming to us, and small startups, if they can outsource some work, they're outsourcing to us.
Okay, thank you.
Great. Any other questions? Okay, then thank you very much, Pan Andrei, and we will move to the next speaker. And next speaker is me. I try to share my screen. Do you have it?
Yeah, we see.
Do you see it properly? As one
big and one small.
Okay, then it's not properly. Mhm.
Yeah, but not full screen.
Not full screen. It is bad. Okay, stop sharing and we'll try again. It's the same.
Yes.
Very bad.
No, it's okay. We can see.
I have two screens here and maybe it is better.
Yeah, now it's okay.
Okay, good. So I'm representing the German company Nomad Bioscience, which is located in the city of Halle. Also, I'm Ukrainian, graduated from Kyiv State University Biological Department, and I worked for a while at Ukrainian Academy of Science, Institute of Cell Biology and Genetic Engineering. I work in the field of plant molecular farming. In Ukrainian, it is sometimes translated as molecularne землеробство. This is a relatively new field which appeared approximately 30 years ago. It's another use of plants. From millennia, people used plants for food, for production of fiber, for clothes, for construction materials, for medical use. And we are using plants as a platform for production of foreign recombinant proteins to be used for different purposes. They are technical reagents, nutritional products, industrial enzymes, various protein-based materials, and also pharmaceutical proteins. Our company was founded in 2008, and before that we started as another company, Icon Genetics, which was founded in US, in New Jersey in 1999 by Professor Yuri Gleba, another Ukrainian who was also the former director of the Institute of Cell Biology and Genetic Engineering, where he worked, and he is currently the CEO of Nomad Bioscience. Our company has also the subsidiary in Vilnius and one spin-off, Number One Biotech in Germany, which has the subsidiary in Spain, which is called Number One Spain. We are focused on research and development at Icon Genetics, and further at Nomad. We developed a proprietary platform for production of recombinant proteins in plants. They are registered at Magnicon and Nomadic Systems. And currently, Nomad is focused on the development of products based on antimicrobial proteins for food safety and medicines, sweet proteins to be used as food additives, and also lectins as antivirals. I will give you short information about three types of these products, just to give you the flavor of that what we are working on. First, antibacterial protein biologics for medicine. That group of antimicrobials which we work on are antimicrobial proteins, bacteriocins. This group of antibacterials was discovered approximately at the same time when classical antibiotics, small molecules, but they were somehow neglected by the medicines because they are proteins, they are less stable, they are more narrow specific, and the success with classical antibiotics was great. But nowadays, when the problem of multi-drug, pan-drug resistant microorganism became more and more serious, research community came back to this group of antimicrobials. So we work with bacteriocins. Bacteriocins are ribosomally synthesized proteins which are produced by gram-negative bacteria. They are active against same or related species of bacteria. And bacteriocins produced by E. coli, Escherichia coli, are called colicins. Produced by Klebsiella are klebicins, by Salmonella salmocins, by Pseudomonas pyocins. If you look at colicin-like bacteriocins, they are protein molecules in size between 17 and 80 kilodaltons, and they're composed of 3 functional domains. The N-terminal domain is responsible for the translocation of colicin molecule into the target cell. Central receptor binding domain is responsible for the interaction of with extracellular receptor of bacterial cell. It is needed for the penetration of bacteriocin molecule into the target microorganism cells. And C-terminal domain is so-called cytotoxic domain, which exerts antimicrobial activity. It can be nuclease activity, in such bacteriocin destroys nucleic acids of target cells. It can be pore-forming activity. In that case, bacteriocin is inserted into the inner membrane of gram-negative microorganism and makes that membrane leaky. And third group of bacteriocins have inhibiting activity for the bacterial cell wall. They inhibit the biosynthesis of murine. In the producing cell, bacteriocins are usually co-expressed with immunity protein, which blocks the toxic activity of bacteriocin against the cell producer. We perform the expression of bacteriocins in plant cells. There are some limitations with large scale and high level production of these proteins in original bacterial cells because of their toxicity. They're not toxic for plants, and you can see here the large protein band which is present in all samples. It's rubiscolate subunit, plant enzyme, which is very abundant, and all other bands which are marked with red asterisk are bacteriocins. We use different discovery platforms to find the right molecules which we would use for various purposes. We first mine the natural diversity of existing bacteriocins, searching through sequence databases. Then we perform the shuffling of domains, functional domains derived from different molecules, and combine them together. Then we use also fusions of bacteriocins to membrane penetrating peptides to improve the activity against intracellular pathogens. We also use fusions to improve their plasma stability. In that respect, we follow traditional approach, traditional selection process of preclinical studies, and we form initial pool, and then we apply primary, secondary, and tertiary screens. We analyze the expression of these proteins in plants, their in vitro activity, their stability upon the storage, their in vitro activity against libraries of pathogens, including multi-drug and pan-drug resistant ones, the activity in body fluids, pharmacokinetics. We analyze also the cytotoxicity. We analyze their synergy with other bacteriocins, and standard of care antibiotics. Based on the primary screen, we identified one colicin, colicin U, which looks like most promising for many applications. Among colicins which were tested with libraries of E. coli pathogens, it showed broadest and highest activity. This protein forms, it's porine, it forms spore in target bacterial cells. And one of possible indications, one of possible application of that bacteriocin is a control of bloodstream infections. This slide shows you some numbers concerning the episodes of bacterial blood infections in the world, in the European Union, in USA, and you can see from these numbers that it's quite a serious medical problem. Usually blood infections are secondary. There is some other source of infection in the organism, and then that infection, when it's not cured, penetrates the bloodstream, and it can bring to sepsis and to the lethal situation. And one of most prevalent bacterial pathogens which cause bloodstream infections is E. coli, Escherichia coli. We tested the, we studied pharmacokinetics of colicin U with different animal models, with mouse, rat, and pig. And we have found that the half-life of this molecule in the bloodstream, depending on the dose, lasts between 40 plus and 50 plus minutes, which is significantly enough to perform some, to show some antimicrobial activity. We tested E. coli, we tested colicin U also in for the fish, for the efficacy in animal models. Here you can see the results for murine model. Animals were first infected intraperitoneally with E. coli, and then with the interval of 2 hours, they were injected with bacteriocin. After 9 hours post infection, they were euthanized according to the European rules. It's difficult to have the longer study of the study because of ethical restrictions. And you can see here that colicin in dose 3 and 30 milligram per kilogram body weight eradicates bacteria from the bloodstream. We performed also survival studies in mice model. It was done in Taiwan. And you can see here that animals were treated and monitored for 7 days post-infection, and doses of 10 and 30 milligram per kilogram body weight completely protected animal against E. coli infection. This data were obtained for immunocompetent mice, and in case of immunocompromised mice, we have similar results. We are on preclinical studies with this potential product, and we are performing now other studies. And first of all, the analysis of the synergy of colicin U with standard of care antibiotics and other bacteriocins. Other use of bacteriocins is for food safety. I will show you the data only for one pathogen here, Salmonella. You know that this is quite a nasty pathogen which causes also there are a lot of cases of infection with salmonella, and in many cases it also gives quite bad consequences, including deaths. And at this moment there is no efficient control on salmonella contamination in eggs, poultry, and pig meat, and at least in the United States, according to CDS data, about 25–30% of meat which is on the market, which is sold, is contaminated with salmonella. We tested the number of salmocins, bacteriocins from salmonella, and we have found that two of them, SAL-E1A and SAL-E1B, are broadly active against that salmonella strains which we tested. 109 strains representing 105 serotypes of Salmonella enterica, subspecies enterica. We tested the use of these products for the contamination of various food matrices, poultry meat, beef, tuna meat, whole eggs. On this slide, you can see some data generated in these experiments, and as you can see here, the use of salmocin reduces the bacterial load significantly, and sometimes it also eradicates bacteria completely. We have some regulatory approvals for our bacteriocins to be used for food safety, for food decontaminations. These approvals were obtained in the United States following a so-called GRAS regulatory pass. GRAS is an abbreviation for the phrase generally recognized as safe. This regulatory pass exists in the United States and few other countries, and it means that the product which get this designation is safe for the use, and it is broadly generally recognized, and one can commercialize and use that product having GRAS approval. Our bacteriocins got the status of food processing aids in the United States. It means that their technical effect is not present in the final product, but is shown only during the processing of food product. We are working now on such approvals in other countries, including European Union, Japan and China, but it's still in the future. Another project is on sweet tasting proteins to be used for food industry. You know that we overconsume sugars, and it brings a lot of negative consequences, including obesity, overweight, diabetes, coronary diseases, and it would be very nice to decrease the consumption of sugars, and the approach to follow is using some other sweet compounds which would be not as calorie rich as sugar but provide the same sweetness. We propose to use recombinant palmitin-2 protein. This protein is naturally produced by African plant, Katamfe, and it is already used for a while, for several decades in the food industry as a sweetener and taste modifier. But the production of palmitin is limited because the raw material is collected in the wild nature, and the resources of Katamfe fruits are limited. we propose to use recombinant thaumatin made in plants, particularly in the seeds of tobacco, transgenic seeds of tobacco. Again, for this product, we obtained GRAS regulatory approval to be used as a sweetener and a taste modifier from FDA in the United States, and also from FEMA, it's a Flavor and Extracts Manufacturing Association, American organization. And we also registered trademark for thaumatin as Tauma. In ancient Greek, it means wonder or miracle. If you look at this map, you will see that FEMA and FDA GRAS status is recognized in many countries, although the jurisdiction, primary jurisdiction is United States. But if you go to other countries, you have to apply for their approvals, but having a GRAS status from US, you can get national approvals in many other countries much easier than without it. Here you can see some expression data, tobacco seeds, protein isolated from tobacco seeds, and this fat band is thaumatin. Here are native proteins from tobacco seeds, which has to be removed. And final purified product looks like that. Here, the single high peak for purified thaumatin. In last 3 years, we performed pilot studies in a greenhouse and in field in Spain, in Extremadura, where we have some contracts with local tobacco growers, and it is a step towards the industrial manufacturing of thaumatin. We grow tobacco plants in greenhouse, partially in fields, and we collect seed capsules, dry them, we clean seeds. They look like that here. They're quite small, like poppy seeds. And then the seeds are used for the purification of final thaumatin product. Thank you for your attention.
Thank you, thank you, thank you.
And I welcome you to ask questions.
May I start?
Yes, sure.
Todek, thank you very much for a very interesting presentation. I know your company is, I think it's the best one in the field. And it's very interesting to see the level of expression. It's extraordinary, like some of the bactericins show maybe 40 or 50% of crude protein, and that is the recombinant protein. So what was the system? Was it transient or stable transfection for both for thaumatin and thaumatin in seeds? It's most probably that is already not transient, but stable transfection, okay?
Thaumatin seeds, it's a stable transformation, stable transformants, seed-specific promoters. And the yield in that case about 6 grams of thaumatin per kilogram seeds. And the gel, which was shown at the beginning for bacteriocins, that proteins were expressed using transient system in leaves, not in seeds, in leaves. And in that case, plant viral vectors were used, MV-based plant viral vectors, Magnicon system. We have also transgenic lines which produce bacteriocins and thaumatin upon ethanol induction. You grow that plants in a greenhouse, and then you spray them with 10% ethanol solution, and they start to express protein. In that case, also viral vector is used, but that virus is silent before you treat plants with ethanol, and the expression level is comparable with transient system.
Really good, very high technology. Thank you very much.
And we see a hand from Dr. Henschel, Diana.
Oh, I thought that was really interesting. I mean, all of these have been really interesting, but I mean, I look at this and especially the antibacterials, and I wonder if you could use it in food. So if without processing the plants, but if it were allowed to be or able to be added to food products, it could be distributed much more cheaply, pretty widely, and still act as an antibacterial, and especially third world countries that are having trouble getting medicines. What are the possibilities of that?
We do not, at this moment, we do not have the permit to do it. And it depends, but technically it depends on the food product. These proteins can be unstable in certain food products during storage. So it should be decided on the case-by-case basis. But theoretically, it's possible, yes.
Even if the plant were dried down and then transported dried, would that make it more stable? Because you
Oh.
can still add it to cooking as a dried plant, right?
Cooking?
Yeah, in food. I mean,
Yeah.
the whole point would be to be a good way and a cheap way to distribute
Yes, but
antibacterials in places where medicines are hard to get.
yeah, we would recommend in such use using purified protein, not plant biomass, also because it is produced in Nicotiana, and Nicotiana contains toxic alkaloids. That's why it should be. That's why they should be removed, and proteins have to be purified. They definitely can be transported in the dry form, in lyophilized form. In that form, they are stable for ages, and then reconstituted with the water and used for certain purposes. Yeah.
Part of what is interesting about the plant-based manufacturing is that it could be sold as the seeds for the plants, right? And then it's cheaper, it doesn't go through the extra processing. And I mean, the Nicotiana is toxic because it's smoked. And the alkaloids, it depends on what they're, how they're treated. So I wonder if there's a way to use it. I'm just trying to think of a way for getting,
Yeah.
when you're in Africa, it's hard sometimes to get medicines, especially in the outback and stuff.
Exactly. Nicotiana is toxic, but only green parts are toxic. Seeds are not toxic. They do not contain alkaloids, and even in Europe, tobacco seeds are approved as a feed for animals. That's why, yeah, in case if some useful protein, antibacterial protein is produced in seeds, one can use ground seeds directly as antimicrobial product.
So I just want, I mean, that's the kind of thing you could approach the Gates Foundation with.
Yeah.
Because they're doing a lot of work trying to get medicines into Africa.
And I came recently from South Africa. There was a conference on plant molecular farming, and I saw that people are very enthusiastic about medicines made in plants because they believe it's a cheap, inexpensive alternative. And they're very often in Africa, they received such medicines or vaccines, being not first in the row. Yeah.
Well, I mean, if you could distribute the seeds, it's so much cheaper. And, and they don't have manufacturing necessarily there. So I just think it's a really cool possibility. Anyway, thank you.
Thank you. And
I have a question. I have one comment and a question. So with plant-based manufacturing, only certain product can be manufactured in plants. So we have to keep it in mind when we thinking about plant-based manufacturing. Here in our so-called bio corridor, we had a big company, iBio.
Yes.
And they were trying to manufacture a vaccine in tobacco plants, and they miserably failed and company crashed. So it's not existing anymore. But whenever you're producing, I don't know, these molecules, and they're efficient, then what is the scale of production? And are you trying other plants other than tobacco plants?
We here at our company we work with Nicotiana species, with Nicotiana bentamiana and tobacco, and concerning iBio and Caliber, I think initially it was a Caliber facility in College Station, in Texas. We know that people, and they were not successful. It happens, but it does not mean the platform in general is not successful. There are other cases of failures. In our field, it depends on the choice of product, depends on the things, but it happens also with other platforms.
Yeah, that's true. That's true. So the product is important. Are you trying other plants or not?
So at this moment, no, we do not try them, but other people working in that field are trying. For example, very first plant-made recombinant proteins, which were on the market as reagents in 1990s, they were made by Prodigy company in the United States, in California, in rice seeds. In rice seeds. One can use, some other people use barley seeds. Human growth factor made in barley seeds is commercialized by one company from Iceland. It's used as a component of creams for cosmetics. In general, there is quite a broad variability of plant-based production platforms. Maybe it's even not very good for the field because it needs different regulations, and regulatory authorities are sometimes lost in that variability. Why we use tobacco? Why we use Nicotiana? Because the expression levels are very high. But Nicotiana bentamiana, our main production host, Australian plant, is not good for growing outside. One needs to have relatively comfortable conditions, greenhouse highly controlled, and then you can get a lot from that plant. If you go to the field, then you have to use some more robust plant, and tobacco is like that. Why we use tobacco? Because often also the regulatory authorities do not like if the food crop is used, because the recombinant seeds can be somehow mixed with which are used for food, and there were already scandals in that respect, and some restrictions imposed after that.
Thank you.
Welcome. Okay, thank you very much. And then I will introduce the next speaker, Professor Diana Henschel from Indiana University in Bloomington, United States. And the title of her presentation is Climate Resilience from the Ground Up, Encouraging Climate Adaptation One Building at a Time. Welcome, Ms. Henschel.
Hi, guys. Okay, hold on. Let's see. All right, so I went from lab to field to system modeling to Igor smiling.
I'm glad to see you. I'm glad to see you.
It's good to see you too. I wish it was in person.
Okay.
All right, so lab to field to system modeling, and then I started shortly before COVID not a good time to start something, working on climate resilience and working on it from the community level, trying to figure out how to best address the really growing problems associated with the extreme weather that we're dealing with. All right, so climate change, as everybody knows, whether we acknowledge it or not, is worsening. In the United States, we have a big group of people that seem to deny its existence. Don't know how. The weather is more extreme. It means that impacts can be very, very extreme locally in a very different way. And impacts can be like, even from, you know, neighborhood to neighborhood, you can have completely different kinds of experiences. And currently, at least in the United States, and as far as I can figure out in Europe as well, most of the climate adaptation work that's going on is top down. And that's not really as helpful for the people that are stuck dealing with their very local situations and very local problems. So from the municipal top-down perspective, it's what are we doing from a municipal perspective to be ready for it, but that doesn't really address what's going on at the individual level. So this work started to focus on how to look at increasing resilience and increasing adaptation to make individual homeowners more prepared in case their homes are more vulnerable. So we started to look at how do we quantify both the and assess metrics that are kind of more local, and then how do we integrate in planning measures to help people actually figure out what they can do for their house in their house situation. All right, let me close this part down. Sorry, guys. All right. So most current efforts focus around critical infrastructure, and usually it's from a top-down planning perspective. But what's happening tends to happen. I mean, when you look at the whole city level, sometimes it's whole cities, but sometimes here it's literally one neighborhood is flooded and another neighborhood is just fine. So we can't really do it, and you don't get the same kind of feedback when you're just looking at it from a municipal and higher up perspective. So we started by looking at critical infrastructure and decided that critical infrastructure was too complex when you look at it from the national federal level. So in the US, we have 16 critical infrastructure categories, and in Europe, you have only about 10. And they're a little more collected, I guess, but even in Europe, it's still focused on the national perspective because it's all based on how is it going to be regulated. And you can't really work on metrics from a national perspective when you're trying to look at things from a very local perspective. So therefore, and the other problem is in an emergency, which is what these weather things are causing, is these crazy emergencies. You can't even translate how the management from the federal infrastructure perspective even translates to the immediate needs. And so that's where our work kind of started, was trying to translate down to the local level. So we started by looking at what happens from an emergency from a very local perspective, and the communities and the individuals are thinking about what's working, what's not working, what they have, what they don't have, and how they survive is really what it comes down to. And fortunately, there aren't that many people that die in floods, but there's a lot of people that get, well, that's not totally true. When the floods are as big as we had Katrina, or you had in Belgium, which is not in Ukraine, but still, there's actually a fair number of people that die, and they don't necessarily die from dying in the floods. They die because you know, things like they don't have their critical medicines when they're there, or they get washed out from a road when they're trying to escape. Anyway, so everything kind of needs to be translated down in order to start thinking about things from the individual survival level. We also had, at this point, COVID was going on while we were working on this. And even in the United States, it was recognized that the critical infrastructure categories weren't really meeting the needs and weren't matching what even our country considered to be critical workers. So for example, education was considered to be a critical worker, so teachers. Childcare was considered to be a critical worker, and clearly those are not part of critical infrastructure. And yet, if you're thinking about things from a survival level, think about being an individual in a house facing a flood and having to figure out where your kids are and collecting your kids before you evacuate to another place that's like significantly farther away. So we started by revising the national critical infrastructure in our country to a community-focused critical infrastructure. We did it first by combining categories, and it's from what is perceived at the individual level. So we have 3 energy sectors at the national level. That makes 0 sense from a household perspective. So that's one energy category. We translated agriculture to food and what it means to actually survive and have food when you may not be have access to food, or certainly restaurants or food chains or anything like that in the near future. So just the concerned issue of food. Similarly, water. All communication got combined to the internet. All in our country, we seem to have a lot of critical infrastructure associated with jobs. We put finance together and we put transportation together. And then we added what we consider to be part of real critical support functions from the immediate individual level. So that's food, emergency shelter, and childcare and education, which in the immediate always is the last thing to even be worried about, but is from a where are my kids perspective is a big issue. So this is how we combine things out. And then the last category of education and childcare has never really been a part of the national critical infrastructure in this country, even though childcare workers got declared critical workers during COVID. Teachers, it varied by state. So then we started by figuring out what was going on from the individual perspective. So we put together a survey that went out to the Great Lakes areas, because the Great Lakes is one of the areas where we're getting increased flooding. And so it looked at risk history and it looked at risk perception, just trying to quantify what was going on in terms of threats, their household perspective of vulnerabilities, and their community perspective of vulnerabilities. Also, again, still focusing on this revised critical infrastructure categories perspective. So overall, despite the fact that flooding is probably one of the biggest concerns, erosion is a greater concern, and that's because we have, especially in the Great Lakes, shoreline houses that are literally falling into the lakes as a result of, you know, a single storm damage and a house could go. When we look at safety type of measures, 2/3 of the respondents, and this is like, so about 900 people started the survey. About 200 either didn't finish or were not part of the correct watershed. So if you were out of the watershed, you weren't allowed to finish the survey. 2/3 of people within the watershed that responded have no public transport within half a mile. We figure half a mile to be quick moving, and even people that have disabilities can get someplace within half a mile in a reasonable period of time. And that for us was about 15 minutes. 80% of respondents, there's no disaster preparedness training or experience, including in their communities. About 20% have no community-based post-disaster programs at all. That's a lot. Only about 50% of the respondents have any sort of emergency kit, which means 50% don't and are not prepared at all if anything happens. And only, and it's clear that even in the last 5 years, so this is 2019 to 20, so 2015 to 16, there were 10% had shelters and food sources that had to close due to storm damage. So clearly things are being impacted and people aren't being prepared. So we started focusing on how do we get individuals to be prepared to survive what is effectively that period of hours to even days where even municipal help might not get to you. So we developed a guidance, and part of this talk is an invitation if people want to either look at this and see how it applies to them or want to start working on it. I realize you guys are all lab-based guys, or not everybody, but people I've heard so far. And I know there's some community resilience work going on in Europe. So anyway, the community resilience guidance is trying to get people to look at things from a vulnerability perspective. We use the strengths, vulnerabilities, threats, and opportunities approach to assessing. Well, it comes from assessing organizational problems and how to do strategic planning in organizations. And we adapted it down to individual homeowners at this point. So it's to help get individual homeowners to identify their vulnerabilities and their possibilities for resilience. We developed a toolkit with linked decision support tools, and I'll show you that in a second, at least one of them. We have, the goal is to get people to use the guidance to start preparing their homes and their neighbors' homes for future extreme weather events. And then our next steps are to do the focus groups to both improve the guidance and also adapt it for apartment dwelling, for small businesses, and for other like non-US type communities. So just to take you through a little bit on the decision support tools. We have two different types of templates. We have an Excel template, and this is the Miro template that helps take people through the strengths, vulnerabilities, and opportunities in response to the defined threats, which are, in this case, storms. We would also then start to develop a guidance for droughts and wildfire later, because those are also issues in the States. And this particular tool starts taking you through in a kind of brainstorming way, and then allows you to go from that to prioritization of your actions. We also have a spatial database that's at this point just for the Great Lakes watershed, and it allows you to superimpose a whole slew of different metrics, socioeconomic and other types of metrics, all of the critical infrastructure pieces and metrics for the critical infrastructure, so that you can identify what's vulnerable to flooding and what's not vulnerable to flooding. And also we can get it down to the street level, where you can superimpose the flood literally at the street level, so you can use it for planning evacuation routes in case you think you're going to be flooded. The flooding is from Fathom Global. They're in the UK. They've done pretty much the whole globe at this point. And it's based on IPCC assumptions. I think it was at 5, IPCC 5. And with a LiDAR-based modeling. And this can be used, this one shows in this map, we've got some overlapping of flood areas and predicted flood areas. I think this is 100-year flood. We've got 100, 500, and 1,000 from Fathom. And these are different types of shelters. So you can see what shelters are available in the flood zones and which are available not in flood zones in this case. This one just shows hospital access. In the database, we have it at the larger level, everything's averaged down to 5-kilometer grid for ease of analysis and for, honestly, because of how much memory it takes up when you're trying to do processing. And as it is, when it's at the 5-kilometer grid, you can still work it on a regular computer without going to the high capacity ones. So I would love to have partners in Europe or wherever you guys are. If anybody's interested in working on it or looking at it or partnering to see how to adapt to different types of communities, I am very interested in partnering. And thank you. So.
Anatoly, you are mute.
Sorry.
I
Thank you.
will unshare. Give me a second.
Please ask your questions.
Okay, first of all, thank you, Diane, very much that you are with us. May I ask you, it's about, for example, this guideline, more for behavior, for community kind of reaction, or it imply change of infrastructure, I mean physical infrastructure?
So since most of the work here is being done at the municipal level, and municipal level doesn't really address what happens to the homeowner, this is how do you look at your house and where you're living and assess whether or not you're vulnerable to flooding. So that's what the database can help with. And then literally go through your house and kind of identify, if I'm in a flood zone, what in my house is vulnerable? Am I going to have problems with energy? Do I need to change things around for energy, or do I need to have like an emergency kit that's got backup flashlights because my lights are likely to go out? So it literally takes you through the types of critical infrastructure and says, here's the issues. Here's the kinds of things to look for. Here's ways to develop an adaptation. Here's things to think about. And it goes from super cheap adaptations to, putting in backup energy systems, which is not so cheap. So it tries to cover the range and allow people just to develop new adaptations. And the goal is to have everybody in the end look at your own house situation and kind of go, this is what I can do to make myself safer for the future. If you're in a flood zone, this is mostly for flooding and storms at this point.
But does it imply some community support? I mean, financial, or it's more advice?
At the moment, it's individual, but if there's a lot of people that work on it, so my goal is to have like neighborhoods work on it together, and then they can identify what they could do as a neighborhood that might change a situation. So our new developments, for example, have to have storm catchment areas, but the old neighborhoods don't. And so there are old neighborhoods that don't have sufficient storm catchment, and they could develop some within their neighborhood to try to reduce the amount of flooding that they're liable to have. That kind of situation. It's really at this point just a tool to start figuring it out, because most people aren't thinking about it from this perspective. In fact, nobody is thinking about it from this perspective at this point that I can figure out.
Okay, thank you.
Please, other questions?
We will touch with you. We will touch with you.
Just
That's fine.
yeah. Your presentation revived some memories. We had the collaboration with one company located on their border between Kentucky and Indiana. And my first visit, I experienced a tornado there, and one of next visits, the flood on the Ohio River. So that disasters which you showed were really huge. And I've heard the opinion that if more often people would build their houses of bricks, not using other technologies, and they would survive better, at least storms. What is your opinion? What is the reason why people do not use this technology?
Well, I mean, you're living in the States right now, right? So
No, in
it's,
Germany.
oh, you're in Germany.
Mhm.
Housing in the United States is becoming very, very difficult. And the generation that is our kids' generation right now are finding it very hard to get to buy their own house or even buy their own apartment. They're all renters right now. Not all, but many, many more than in our generation. I think the post-war and, you know, many decades after the post-war generation, there was a lot of emphasis on trying to get communities built, and that led to a lot of people getting housing. That was in both Europe and here. All the problems after 2008 collapse has just caused so many financial problems that this generation is having serious problems and not being a renter. So, you know, everything is build as cheaply as possible these days. I can't speak to Europe, but in the United States, it's build cheap. And build cheap is not going to necessarily make you less vulnerable, more resilient.
Yeah.
And so we've got confounding influences that are causing people building in floodplains, people building in areas that are prone to tornadoes. We have more tornadoes happening here. On the coast, there's more hurricanes, there's more storm surge. And they're not kind of thinking about the fact that when you build there, it doesn't take very long for problems to occur and it gets to be more expensive. I don't know. People aren't thinking ahead. I don't have an answer.
Thank you very much. Okay, and let's move to the next speaker.
Oh,
We have one more hand, no?
Sorry, I missed. Yeah, please ask your question.
Okay, this is me. I'm Olga Kirilenko. I'm working in Kyiv Medical University. That's why I'm interested in the aspects related to human health and more than survival, like just survival, because health is also survival. Then what about the in the United States in context of this erosion, erosion of ground? Planting of plant, because on your picture, even on your picture, it was no plant around the house going to the river.
So
then,
they're putting down riprap. I'm sorry.
So because as I know, and we can solve several like very global problem in the same time in context of sustainable development, because as everybody knows, then carbon dioxide is one of the global also, and it's as I know, then 2 kilotons of carbon dioxide we have like plus every year. Only I was surprised to see that it's only 4 kilotons. It's very so meaning that our plants they consume every year like around 300 kilotons or 400 kilotons, so meaning 4 kilotons. If we plant more plants, very easy to compensate and to compensate the erosion of soil. For my opinion, then people think about have logic that somebody needs to do this. But in case of that people who live in under such risk, they already need to understand that they need, and everybody needs to like impact to plant some plant at least. Yeah.
So I agree with you. The other issue though is that when you have massive erosion, it tears down the plants, and the plant roots, most of the plant roots that they use are grasses or pretty shallow roots, and those don't protect as well against erosion. The other issue in the United States is when there's drought, the plants die, and that also causes problems with protecting against the erosion. And then in the West and Southwest, there's been huge problems with wildfire. We actually have started to get some wildfires in the Midwest, which is kind of unusual, but it's happening. And there was a huge Texas wildfire. I think it was less than a year ago. So I think it was this winter. So, and then wildfire also causes major devastation to plants. And then you get, when you have storms on top of wildfire, you get huge erosion problems and mudslides that happen. So, and the mudslides can be very deadly, and they can take out whole neighborhoods. So, and that actually, there's probably more in California, there's probably more death from the mudslides, from literally houses getting covered by mud, than there are from the floods themselves, because you can escape from the floods. But when mudslides happen, it happens like super quickly, and it's like the side of a mountain kind of washes down and covers everything below it. Imagine like lava happening from a volcano, except for it's mud. Anyway, so, and those usually, the mudslides happen after there's been a fire. And so, yes, we can plant, and yes, we want to replant, but that's not going to protect totally against these repeated, fast happening, one on top of the other within weeks, or, you know, maybe a few months, kinds of climate extremes.
Thank you.
Thank you very much. Let's move to the next presentation. Our next speaker is Gustavo Enrique Denzintonoli, also from American continent, but from Southern America, from Brazil, Federal University of Lavras. His talk has a title, Challenges and Applications of Cellulose Nanofibrils in Different Contexts for Sustainable Development. Welcome, Mr. Tonoli.
Hi, could you hear me? Yeah.
Yeah.
So thank you very much for the invitation. So I would like to share my presentation here. And so I'm trying to find this. So what's the best way to share here? What do you suggest me?
At the bottom of the screen, you have the green button, Share Screen. And then you should press it and select the window which you want to share.
Okay.
You are probably Teams user, yeah?
Sorry?
Are you Teams user?
Exactly. Or Google Meet user. I not use
so
much.
You move your cursor to the bottom of your screen, and then the menu bar is opened. And you see here the green button, Share Screen.
Yeah, after that, I didn't find what my window. That's the problem. I don't find my window here.
Is it open?
Did you see here? Did you see that?
No, not yet.
No. Yeah. It's not appearing for me the option of my. Mhm.
Usually, if you click on that green button, you see several windows. Which
Yeah,
are
but the window I want to share is not appearing here. So
is it opened?
it's
Is it opened?
my PowerPoint is not appearing here. So I can't find it. Sorry. Desktop, whiteboard.
Gustavo,
Microsoft.
please,
Yeah.
you can use screen, just up and in a row, up and left, the most up and left and screen, and then we will see everything what you have on your screen.
It doesn't appear for me. The screen.
Okay,
here.
maybe other option is just to send me now to your presentation to your email, and we will share it from us.
Yeah, it's a good idea.
Yeah, just forward it to my email.
But don't worry. So
don't worry.
It's okay. Okay, yeah. Actually, we may change the reports. Maybe now we
No, it's
may...
okay.
Yeah.
It's okay. We will start now. We will cope with that.
You got it? Yeah. Great.
usually use Zoom for our business purposes. And if someone sends the invitation in Teams, we have the same problems. It usually takes 15 minutes to adjust everything.
For the first time, yes. But
That
Gustav,
is why my proposal, we may listen to the next speaker, and after that we will return to Gustavo. No?
okay.
It's okay?
Good. But Gustavo was saying that he is restricted in time.
It's okay. Let's try.
If he has it already, he can show, he can share his.
No, I don't have it yet.
You don't have it. Mm-hmm.
No, it's attaching. It's a big presentation, so
Too big. Send them.
So sorry, people. Just a couple of minutes. It happens.
Yeah.
It's typical problem.
Yeah, why it don't appear for me?
Yeah. No, it's okay.
Don't worry.
You should close your PowerPoint and open it again. Sometimes it helps.
Okay, I will do that.
So you mean you sent it to me? I didn't get it yet.
No, I sent.
Yes.
Already sent.
No.
Yes?
No. Yes.
Okay.
Did you receive it?
Virus scanning. Now we will get it soon.
It's good that you have no updates on your computer at this moment running.
Hopefully.
It's loading. It's moving. Download. Download anyway.
Are you in Brazil now, Gustavo?
Sorry?
Are you in Brazil at this moment?
Yeah, exactly. I'm in Brazil.
Which time? Which time do you have now?
Now is 10 a.m. Yeah.
It's a big presentation, and it's loading, and almost half is loaded. So it's okay. We will have it now, soon.
Sorry for that.
No problem. Don't worry.
And what's about temperature right now outside, Gustavo?
Oh, no, because I'm here. Here I'm in Belém, in the Amazon forest. So here is very hot. I think around 28, something like that.
Great, just great, great for me.
But no, but I live in Minas Gerais. It's more in the south of Brazil, so there is not that hot. But here in Amazon forest, so it's very hot.
Can you see
Oh, great.
Gustavo's presentation now?
Yes,
Full
yes,
success.
yes. Okay.
Full success. We have it now.
Ura. You
Oh,
are in
great. Thank
Belém.
you.
Wow. I
Yeah,
want to go to Belém.
Yeah, Belém is
Okay,
very
okay,
nice.
we are all
The coast.
attention.
Okay, great. Thank you very much for this opportunity. So I will show you a little bit of our work we do here in Brazil. Yeah, so it's a little bit of contribution to more sustainable materials. Yeah, what we do here. So my name is Gustavo Tonoli. I'm working in the Federal University of Lavras in Brazil. So I will present you. Could you go to the next slide, please? So I will show you a little bit of our university, of our labs. Next. So here we are in the South America. As you know, Brazil is here, and I'm located in Minas Gerais state. That is a state located around near São Paulo and Rio de Janeiro. So it's a big state. Go next, please. And Lavras is located in this state. This state is the state of mountains. So we have lots of mountains here. Next. So the best, we can go fast, please. So we have here the most historic places in Brazil. So these are the places where Portuguese came to take the golds and diamonds. So those historic places, because of that, they explore a lot the gold in this place. So also this state is the state of the coffee. So we have here the best coffee of the world. Also the cheese are a specialty of this state. Next, please. The coffee grows very well in high place. So the best coffees are originated from high place, high altitude. Cheese also is one of our specialties here in this state. Go, please. Next. Next again. Also, this is the state of cachaça. Cachaça is the beverage, very famous beverage in Brazil. Please go the next. And so here we are located in Lavras. Lavras has this university, the Federal University of Lavras. So we have around 115 years, and it's mainly dedicated to agricultural studies, studies of animal science also. So it's very agribusiness university. And I'm located in the Department of Forest Science. So we work a lot with wood technology. So we are located in the wood labs. Next, please. We have several professors working with us on biomaterials engineering. So we have a graduation program here with several professors working on these topics. So, and my lab is the lab of forest and nanotechnology, where we work mainly with wood-based materials, cellulose-based materials from the forests. So we try to find some sustainability with the development of new materials. Next, please. And so since 2010, we are working with microfibrillated cellulose. That is the wood-based wood derivative from wood, material derivative from wood. So we are working on this to find applications of in different types of materials. And I will show you a little bit about what we are doing here on this topic. So we can do this cellulose microfibers from products from planted forests. So we are a huge producer of eucalyptus pulp, craft pulp that you use it for paper production and packaging production. So we work a lot with this planted forest. But next, please. We can also work with products from agroforests and native forests. We also do a lot of research on lignocellulosic materials from these ecosystems. So also we always say to the importance of trees to the world. So you know that. Go next, please. So also show you the first topic that we work a lot is the improving of the construction of commercial pulps. As you know, most of the pulps for paper production comes from wood. We work here in Brazil a lot with eucalyptus and pine. So we are working in collaboration with companies to help them to deconstruct these fibers, these lignocellulosic fibers, to obtain a material like this gel, show it in this picture below. So we can use this gel for different applications that I will show you. But we are working a lot in the improving of this the construction of the commercial pulps to do this with less energy. We can go, please, next. So we can, it's a challenge for the industry to obtain this cellulose nanofibers with low costs. So we are working on that with chemical pretreatments of these materials, of these cellulose materials, to obtain different types of microfibers or nanofibers that can be used for different applications. So the challenges are this, as I show you. We have industrial infrastructure. We already have in Brazil industrial infrastructure for production of cellulose fibers, but we need to improve the obtention. We need to use less energy to obtain these MFC fibers. So we are working a lot with this. Also, a challenge is the definition of the protocols for characterization of this material. So next, please. Here are some of those challenges. So to obtain these microfibers, we work with sharing force. So this is a lab equipment that we use to obtain them. So we pass our pulps through this equipment that do some sharing force, and then we obtain this gel-like material that is showing here below. It's a composite of lots of very thin fibers. So when we dry this, we obtain a very, very strong material. Go, please, the next. So we are working a lot with this, with the companies. So we have lots of Brazilian companies that produce cellulose pulp and paper. So we have obtained different pretreatment solutions to improve the construction of the fibers. So one of that is using alkaline solutions. So we can save around 40% of the energy when using this pretreatment we developed in our labs. Next, please. Also, we do some pretreatments with abrasive minerals that we mix with the cellulose fibers to pass through that sharing force I said before. So also we obtain a reduction of the energy with these pretreatments. And also next, also the use of enzymes. Next slide, please. Also use enzymes. One more. More. Next. Yeah, so enzymes also, one back, please, one back. Enzymes also, we are important, yeah, here. So enzymatic pretreatments, we are also developed some to improve the construction of the wood materials of lignin cellulose materials. So we can find some enzymes that are able to improve the construction of the wood fibers. So we can, for example, in our works, we find 40% less energy consumption using these enzymatic pretreatments. So go ahead, please. So also enzymatic are very sustainable because we don't need lots of recovery of the reagents. Also, other topic we work a lot is the look for alternative fibers for this obtention of fibrous. So go ahead, please. We are working with different types of vegetable fibers like banana, coffee husks, jute, sisal, wood residues, bamboo, different residues from agro-industry. So next, please. We work a lot with these residues looking to aggregate, to upcycling of these residues. So we have lots of residues from agro-industry, and these are some of the challenges that we are working on. So looking for have some MFC from these residues. Please go next. So also the procedure is the same. So we can use residues that has a little bit of cellulose to obtain this nanofibrous gel, this brown gel showing here in the right. So when we dry this gel, we obtain like a film, very nice film that can replace, for example, some types of plastics. Please, next. Other examples are some cactus. We work a lot with some cactus to produce this nanofibrous. Go, please. Also some types of some other residues like wheat and oat straw. We do a lot with this. Next, please. So it's possible to obtain like coatings that are able to improve the shelf life of fruits, for example. So these films, normally we do these films and characterize them to understand the properties of those materials, and trying to always to improve the performance of the film. So we can see in this slide the improvement of the film when we add these microfibrous. So in the first graph you can see that the improvement of the strength, tensile strength, when we add nanofibrous. Please go. So we also the one of these applications are the coating of fruits to improve, to have more shelf life, for example. Next. So they are able to give like a barrier to the fruit. Also, we work a lot with starch, and so starch with nanofibrous are able to originate like films like that, very transparent. So in our studies, we look for obtain films like those to apply as packaging, for example. So in this study, we find like 25% increase when adding cellulose microfibrous in the composition of the films. also an increase of 20% of the barrier properties. So our aim is to apply these films on different materials, as packaging, for example. So also we work a lot with residues from Amazonian woods that are cut, and as always we have some residues. So we always are looking for application of these residues, upcycling of these residues. Next, please. Here are an example of the obtention of the film. When we have this cellulose in nanoscale, we are able to change the optical properties. So we can see here in a paper, a normal paper in the left side, and in the right side we can see the transparency. That means that we have the cellulose microfibers. Next, please. Next, one more, please. So we work here. I will show you some of examples of the application of these nanofibrils as paper coating, for example. Go ahead. Next. So as we know, we have a problem with the plastics from petroleum based. Yeah. So we have lots of these plastics that are not that don't degrade in the environment. So one of our objectives is to replace those fossil materials and obtain, for example, sustainable packaging. So we are working in collaboration with some Brazilian private companies for improving those paper packaging. As you know, this paper package has lots of advantage, but they are not able to for example, to have a barrier for moisture, or sometimes when the mechanical resistance is also not that good. And so what we are doing here is the incorporation of these microfibrils in the formulation of these packagings, also sometimes with mixture of other biopolymers. I will show you some examples. Go, please. So as you probably know, we have different types of packaging materials. We have packaging that are tertiary, that just to reception, transport. We have secondary packaging that is to distribute, and the final one is the primary packaging that contain the product. So we are working on to improve this type of packagings. Our dream is to use the MFC, the cellulose microfibrils, to have a high barrier to this packaging, high barrier to O2, to water and O2. So we can have packagings that are similar to the plastic ones. That's our dream with this project. Please, next. So then we can improve the shelf life of the products when we have a high barrier. So that's what we are looking for with the companies. Go, please. So here is an example of one of these applications. So Klabin is a big company here that we helped to launch this card paper, and that has a little bit of this cellulose nanofibrils. So they are able to produce like thin cardboards when using this MFC, and also improving the properties of the cardboard when applying this MFC. So that's a successful example of what we did in collaboration with some companies. Also here is another example. Fiberlin is a company that also produce this. They work a lot with this. We didn't with them, but this just show another example that is worldwide used. Go please, next. So here is some examples of the packages that we already use in our days, yeah, but they all of them, yeah, mainly this Tetra Pak box, yeah, they use some aluminum, some petroleum-based resins also to have this good performance. So our objective is to replace this petroleum base with this MFC. That's what we are working with the companies. So here is an example: the bags for coffee, for example. So this bag is normally used to export coffee. So they have normally a little film of plastic inside. So what we want to do here is to replace this fossil-based plastic with our film of cellulose nanofibrils. Go next. So we have a lot of challenge. So that's what we are working on. So trying to solve some problems. For example, NFC are very hygroscopic. So we work on research to improve this issue. Also, when we go to industrial application of polymers, we have lots of details about how it's applied. So we work on to solve those problems of the industry. So also, for example, the viscosity of the polymers, the amount of solids of the suspension. So all of them are challenges that we work with the companies to trying to improve those issues. Next. So here are some examples of the application of this film. So we can do this in different ways. So like bar coating of the paper or spray coating. So those are some technologies that are already used in the industry. So we work on the adaptation of these technologies to put our MFC, our nanofibrils in the composition of these chemicals, of these resins. Next. This is just a other way of applying the coating. Next, please. So we work a lot with the mixtures. So as I said, the cellulose microfibrils are very hygroscopic, hydrophilic. So we need to mix with other materials to improve this behavior. So we work a lot with other renewable source like starch or proteins, natural gums or vegetable tannins or natural resins that are able to improve this performance of the microfibrils. So we have lots of research on that. Next. Also, we work with some additions of minerals, next, like clays that are able to improve also the performance of our cellulose microfibrils. Next, please. So the objective is the overlapping of the layers to find that performance. As I said, that we are looking here for a barrier property to moisture and oxygen that goes to inside the packaging. So that's the main objective of our to obtain this packaging with just with renewable material, substituting aluminum or other fossil-based materials. Go next, please. So we do this in the lab with first applying some coatings in normal papers. We normally work also evaluate after the performance of the papers. So we have lots of standards to do that, standards that are already using worldwide. for example, the water, the vapor transmission, or the contact angle with water. So lots of properties that are important to these applications in packaging. Next. So here are some examples of using. So always the use of MFC, the cellulose microfibers, are able to improve those properties. So we have lots of successful applications. Next. So these are some examples of the characterization we do. COB test is a test that that evaluates the absorption of water of the paper, so we can find some improvement when using MFC. Next. Also, it's very important the oil resistance of the packaging. So MFC was also able to improve this barrier to oil. Next. So here is just some examples. So with MFC, we find the maximum barrier with barrier to oil. Next. Also, some improvements of paper printability. So as I said, nanofibers are very, very thin fibers. So when we apply this on paper, we are able to find like a very smooth surface. So it improves a lot also the printability of the papers. Mainly, as you know, packaging, we need to print information in the product. So MFC also is very good for that, to improve this printability. Next. So all of this we evaluate in our labs. Also here are some examples of applications in paper tubes. I will show you some examples of our work with this. So we have a collaboration with a Brazilian private company that produce these tubes, these paper tubes that are used for different applications. In this case, they developed these molds for concrete. So they need to improve the performance of these molds. So this is something that we are working with this company to improve the performance of those tubes. So there are lots of challenges for that. Yeah, for example, when we put the cellulose microfibers, the viscosity of the adhesive changes a lot. So here they use adhesive, sodium silicate adhesive. So when we put the cellulose microfibers, this adhesive changes a lot. So this is one of the challenge of applying this on paper tubes. Go, please. So we do this. Also, these companies also produce a lot of residues. So what we are doing here with this company is to put these residues to produce the cellulose microfibers with these residues and go and put them again in the process. So one of the challenge is to put these residues in the adhesive again. Go, please. So around 10% of residues in the industry. So the objective of this work is to produce those nanofibers with the residues of the industry. Next, with the same procedures I said before, and go and put again the cellulose residue into the adhesive. So we can see here in this graph the improved when we did that. When we put the cellulose nanofibers in the adhesive, we improved a lot the performance of the tube. So that's what showed that. Other application is the application in, next please, the application in wood panels. So we do a lot of research on that. So we have different objectives here. So the way we put the nanofibers in the adhesive that bond these particles, these wood particles. So we are working a lot on that, trying to put a little bit of MFC in the adhesives and improve the performance of these panels. Also, next. So also we had a lot of challenge here. The adhesive changes a lot when we put the cellulose microfiber. So we work on these issues. Next. So here's an example when we put the cellulose microfiber, we have some increase in the properties. So like the elasticity modulus of the strength were improved when we put those MFC. Next. And the last is the application fiber cement composites. So we work a lot with this type of tiles, and boards are very used here in Brazil as roofing material. So we are working also with the industry to improve those tiles using our cellulose microfibers. So also we have some challenge here because these machines now we need to adapt to these machines that already use cellulose pulp, and normally now we have the substitution of asbestos by other fibers. So that's an issue that we are working with the industry also to substitute the asbestos with more friendly fibers like those that we produce in our labs. So it's a market that's increasing a lot. The use of these boards to produce houses. So the objective here is to put the maximum of cellulose fibers in this type of material, trying to improve the sustainability of this type of material. Next, please. So, as I said, there is a lot of challenge. Yeah, we need to. The cellulose also is alkali degradation in the cement. Cellulose degrades a lot when inside the cement. So it's one of the issues we work a lot. We work with the companies to solve. Yeah, I think that's it. I try to show you some of our contribution to developing of more sustainable materials. So what we want is to at least substitute part of the petroleum-based resins. So we do this looking for the normally with collaboration with some private companies, trying to find the viability of these applications. So there is a lot of gaps that we're still working on to find the solutions. Thank you very much. And if you have questions, I will be happy to answer it. Okay, thank you again.
Thank you very much, Gustavo. Very interesting presentation. We're a bit behind the schedule, just half an hour.
No problem.
We should have the time for one question.
Just maybe one brief. Actually, we are okay with time. We just have additional report. Yeah. Okay, Gustavo, if you, I am impressed with this idea of packaging, application. How expensive it may be? I mean, it's significant increasing of cost. For example, for paper packaging, falsified with these fibers.
So could you repeat, please?
I mean about costs. How expensive the packages will be with these fibers for paper packages? Yeah.
Exactly. Yeah. That's a issue that's very important. So we normally are working on trying to decrease the costs. Yeah. So it's the packaging when have this is a little bit expensive than the normal one. So we are working with the industry, always trying to decrease the costs. But this is like a gamble. Yeah, the industry needs to gamble on this. Yeah, where all of this industry, of packaging industry, are looking for more sustainable applications. So that's a really important point that we always working on, trying to reduce the costs. Yeah, so that's
Okay.
an important issue.
Still need working. Okay. Thank you.
Yeah, it's still expensive. Right? It's still expensive, but
Okay.
we work on that to reduce the costs. Yeah.
Mhm.
Okay.
Thank you.
Any other questions? Then maybe I will ask one question. Probably I just missed that point, and you mentioned it. What are the conditions for solubilization of cellulose?
Yeah. No, normally we don't solubilize them because they are very strong. The fibers. So we use to deconstruct them, because if you want to solubilize, you need some very strong chemicals. So
Yeah.
normally we just use a dispersion of fibers.
But to make the gel out of solid material.
Yeah. To make the gel, we use like a refiner. I don't know if you know the refiner. Normally use it to produce paper. Do you know that refiner? It's like some discs that have lots of runs where in a gap. So we pass the fibers through this gap, and then the sharing force between these discs produce the gel. That's the way to decrease the size of the fibers. So that's the way we use to obtain this suspension of that has the appearance of a gel.
Okay, thank
But
you.
actually, they are not solubilized. They are like very thin fibers. Okay.
Any more questions? If not, then thank you very much, Gustavo. And
Thank you very much.
We are back to you. The next speaker, Günther Pochmann, is from University of Natural Resources and Life Sciences in Vienna, Austria. And his presentation is about sustainability in the food and beverage industry by anaerobic digestion.
Yeah, good afternoon. Thank you for the invitation. Honestly speaking, I'm not working for the university anymore. It's like it was just my research, what I did back then at university. I started a new business, a startup in Vienna. Here we're using hydrogen and CO2 for amino acid production. But my dear friend Yevgeny asked me to give this presentation, also with my background of university. That's why this is more in the direction of anaerobic digestion. So thank you for the invitation, and I think it doesn't fit optimally in the schedule or in the other presentations, but in the morning I was full of meetings, so I was quite busy. So I will start my presentation. And yeah, so it's more or less a medley of my research, what I did back then at the university. I think you can see it right now, the presentation. And yeah, so sustainability in the food and beverage industry by anaerobic digestion. So what we are faced in the industry is like, especially in the food and beverage industry, we have a huge pollution, or we have a pollution what we can reduce because we have a huge amount of organic compounds in our residues, or our residues are only consisting of organic compounds, which can be reduced or degraded easily by organisms or microorganisms, then we have a huge energy demand in any of these processes because we have a huge amount of heat demand as saturated heat. And yeah, for this, we need energy at the same time. And the benefit or the opportunity we have of anaerobic digestion is that we have the opportunity of nutrient recovery and that we are circulating specific nutrients like nitrogen, phosphorus, sulfur, just to be mentioned. And what gave us also an idea what new processes can we develop during during our R&D, because we were always faced with specific requirements which need to be targeted, and that's why we had new findings for new processes. So what is now the demand in the food and beverage industry? We have a huge amount of wastewater because, as you know, we need a lot of water to process all the organic compounds. Then we have, beside the wastewater, pastas and solid products, depending on the industry we are looking for. And then we have the energy production on the other side. So we need electricity, or huge amounts of electricity. We need heat and set steam. So we are in a temperature range of approximately 160, 200, 220 degrees Celsius. And we have the biomethane, which is in specific cases the way to go and gives us the opportunity for the installation of the technology. Then, last but not least, the production of fertilizer. Here we are reducing the demand of nitrogen fertilizer, phosphorus, but also sulfur containing ones. But at the end also the lignin compounds, which have a positive effect on the soils and also the microorganisms which are in our digestate. But everything is limited by the legislation. So depending on the country and the region we are, especially Austria is always very restricted depending on the regions. We have specific requirements how to treat the digestate before we spread it on the fields. So what are the different technologies we were faced within the past few years? We determined that we need different technologies to treat the different feedstocks. One is the classical one coming from the wastewater treatment plants. It's a UASB or GSB. These are sludge bed systems where you only are able to treat wastewater, but with a high loading rate and high efficiency. Then the CSDR, the more classical one, which comes more from, I would call it agricultural anaerobic digestion plant, a fully steered tank reactor, which comes also, or which is used also in the pharma industry, with a different shape of the bioreactor, but it's also a CSDR, or the plug flow fermenter, where you enter with a feedstock on the one hand side and going through the whole system and degrading during this period the organic matter. So we did some research on different bioreactor technologies. So coming to the industries we were faced with in our research, first of all, and the most important one is slaughterhouses, because here we have huge amounts of residues and we have a high nitrogen concentration, which leads to the inhibition of our biocenosis and especially of the methane producing archaea. Then also some research we did work on was on the dairies. So during the milk production, then my PhD was about the implementation of anaerobic digestion in breweries. Here we are always in a competition with farms when you look for the brewer's spent grains. Then olive processing, here you have polyphenols, which might inhibit your process, and this means also be treated or can be treated anaerobically. Then the sugar factories, Brazilians are not used to it, but we have in Europe the sugar beet as a major, or in some plants, there is a major source of sugar. And here we have the sugar beet pulp, which can be also treated anaerobically to produce biomethane. In Brazil, you have the sugar cane, and as a product from the distilleries, the bioethanol, and for example, cachaça. Here they are faced with completely different problems with the fibers, and they are trying to use it for the utilization as packaging material. I'm more the destruction guy. I'm trying to degrade it to bioethanol, to biomethane, and yeah, also a very good application. Then the potato processing industry, chips, and any further fries. Here we have a huge amount of organic compounds which are occurring during the process. And then wineries and the juice factories, these are depending on the season important industries, but if you have a look on the yearly amount, this is quite limited. Coming to some amounts, we had a big European Union project, food and so anaerobic digestion in the food and beverage industry. Here we gathered from numbers for different residues and the amounts which are available in the different countries. To spoil it, Austria, we already use many of our residues, so the 1 million tons a year of wastes are already treated anaerobically to a certain amount and are not available. So, and especially France did a lot in the legislation to use these organic residues anaerobically to the production of biomethane, biogas, or electricity. So in total, we found out that we have more than 30 million tons of organic residues available or already treated. I brought you some examples of the application of anaerobic digestion and how we implemented it. So we did some research, which was very close to the industry or in cooperation with the industry. So one was with a dairy in Wales, the Bergland Milch. They have a feedstock of 360 tons of liquid residue, so it's whey and wastewater a day. And what is also challenging, you see here, the neighbors are very closely. And if we have other emissions, I don't know who of you is used to anaerobic digestion, it might cause some other emissions. Here, one important thing was to make it, to reduce the odor and to have a very good running process. We had a daily production of biogas of 5,500 cubic meters. What they're doing right now, they are producing electricity and heat from the residues, from the biogas. And yeah, the specification is that we have a liquid treatment. So we are pumping from the bottom the whey and the wastewater in the system, and here it's treated and degraded to biogas by lowering the CO2. one example, and to the best of my knowledge, the first one, which is only treating brewery residues, that's the Brauerei, the brewery Gösser in Göss in Austria. It's in Styria. This is the largest brewery in Austria. They have a feedstock of 17,000 tons of brewer's spent grains. For some countries, it's a smaller brewery, but for us, it's the largest one. And additionally to the brewer's spent grains, they are treating yeast and kieselguhr. Kieselguhr they are using for the filtration of the beer. And they have a yearly gas production of 2 million non-cubic meters of biogas. And they're producing electricity, and some of the gas is used in the brewery for the production of steam, so they can call themselves a green brewery. The specification of the plant, and this is where I did a lot of research on, with that we have the pre-acidification. This means we are separating the degradation or the larger compounds and the biomethane or biogas formation from each other into steps. And here in the pre-acidification, they're producing their first amino acids, organic acids, which are degraded to biogas in the next step. Then we have the digestate utilization, because huge amounts of digestate occur daily. These needs to be treated because the farmers trying to get as much as money as possible. And they are sometimes charging for taking the digestate, even if it's still of their benefit to use it as a digestate on the field, the fertilizer. The abattoir in St. Martin, also in Austria, is also worldwide the first one, who is using solely feedstock from the abattoir, blood, rumen content, fats, etc. They are almost energy independent, so they use the gas for the energy supply in the system, and additionally there are some thermal energy. The specification of the plant is we have a very high nitrogen content. How we solved it, we are forcing the adaptation of the organisms. So in the system, we have highly nitrogen tolerant methanogens inside, and thus this process can work constantly. Very nice plant. I once visited in Hungary. This belongs to Agrana, a huge agricultural company from Austria. They are using their sugar beet pulp from the campaign during the season for sugar beet, so only 4 to 5 months. And seasonal, they're adding energy crops. They have a yearly gas production of 160,000 cubic meter. What they're doing right now to reach a completely sustainable process, they are upgrading the biogas to biomethane and injecting this gas to the grid and buying it back during the campaign, so they can claim themselves as a 100% renewable system. One plant from the US, from Oregon. Here they mix different organic residues from the food industry, and they are mixing it with each other, so they are producing electricity and heat from it. They use the heat for the pumpkin seeds drying, and the unique system is also the digester type, which they are using. They have a pressurized system, so they are balancing the water level, and by this they're reaching the mixing of the bioreactor. One problem from Switzerland, here they're just using different kind of residues. They are increasing their slaughterhouse waste, and to a certain amount, they are not reaching the same nitrogen concentration, such as the one in Upper Austria, at Großwörthner. But they are doing pretty well. They're producing approximately 1 megawatt electricity and at the same time 1.3 megawatt of heat. And they're using it for heating stables, which are surrounded in Edelswil, and treating the feedstock of the biogas plant to increase the yield. So which are the specific requirements for these industries? We have the specification of the feedstock. We need to know what we are putting in our bioreactor. The problem in most of the cases is they are trying to feed a not suitable feedstock to a system which is more suitable for another feedstock. So we found it many times that they're adding too much water into a system, then you create floating layers and so on. And so especially if you come to the food and beverage industry, you need to be aware about the specification of the feedstock. So feedstock is not feedstock. Then the retention time, especially if it comes to high nitrogen concentration in your feedstock, you need a far higher retention time. And the industry also tends sometimes to build it as small as possible to make it cheaper and cheaper. But then you have other emissions from the digest state, which is not completely degraded, and then you have this specific other. What you need to take into account is also the seasonality. So how many times and how many months a year does my feedstock occur? So is it, for example, in wineries, it doesn't make any sense to build your own bioreactor because you have the grapes or the residues from the grapes only available for a couple of months, for 2 months maximum, maybe 3 months, but that's it. And storing these feedstocks is very hard. Then the process implementation. If it comes to the heat production, it's always important to implement this in my whole energy system. So the energy demand in my system needs a bit of a focus that I see, okay, where can I make the implementations? So a pinch analysis is here the way to go to see, okay, where do I have the different temperature levels. Then the residue utilization in AD is quite important because you are an industry and they are lacking of fields to use it as a fertilizer. That's why there is always a farmer required who use it as a fertilizer. And with some plants, we didn't manage to find here a consensus between the two partners. It's a pity because it's a good product to use as a fertilizer, but yeah, we are getting a bit better. Then the CHP units, always you need to know what do I produce. If it's CHP, then it's the process implementation comes into account that I have the electricity demand, the heat demand. And if this is not given, like for example, the plant in Kaposchwar, we have the production of biomethane. So they're upgrading it, injecting to the grid, and then buying it back when they need it. And what is very important to mention is the exhaust gas temperature is approximately 400 degrees Celsius, is sometimes required for the production of settled steam. So conclusion, we can use, or we showed that we can use all of these organic residues, but you need a specific process. You need to be aware of the specification of the different compounds. For example, high nitrogen content, Then we were faced with some residues with a very, very high sulfur content. Then you have an increase of the H2S formation in your system and so on. And this is, or you have a feedstock which needs a very long retention time, like for example, fibers. This is also need to be mentioned when you are planning a plant. Yeah, then if you want to go green with your business, this is one option, the substitution of the fossil fuels. Most of the industries showed that they can be independent from fossil fuels. Slaughterhouse showed it in a very nice way. The brewery also showed that they can be almost sustainable. They need some solar power and solar thermal energy to fill the gap to the 100% renewable energy. Then what else comes into play? We have the reduction of transport costs. So we are treating everything on-site the plant, and we are aiming in some parts with a no waste strategy. Yeah, as mentioned before, the successful implementation in several industry is demonstrated already. So we are not in the research phase anymore. For sure, there's still always the potential of improvement, but this depends on every side separately. Yeah, and what you need to know as an industry, you need a specific plant and the knowledge it's required. So the industry has all of a sudden a new business, and they need to learn it, and they need to run this as a add-on to their production site. So I want to thank you, and if there are any questions, I'm happy to answer them. And if you want to have the university contact, Wolfgang Garbauer took over the role as a group leader at the university. I'm working for Archeon. That's why if you want to get in touch with him, don't hesitate to do it. He's giving his presentation tomorrow. So thank you. And yeah, if there are any questions, happy to answer them.
Thanks a lot. Please ask your questions.
We have a hand.
I would like to ask my question, which I was going to ask previous speakers about biogas. Is it possible? Is this possible?
Yeah, ask a question. Yeah, of course.
Okay, so I was after this presentation, I decided still decided to ask this about this aspect, about the safety, again, in context of human health, in context of contamination of environment by the production of biogas. Of course, this methanol and ethanol, it's more clean, like fuel, yeah, after it's used. So for using it's a more like green, more like healthy, but production. In the production, you use, first of all, the fermentation, it's the burning of any of organic matter, especially you mentioned you use sometimes such material as even from animal, like even animal material. Then, and you mentioned containing a lot of nitrogen, yeah? So nitrogen like rich. So that's why, of course, carbon is used for the production of methanol, ethanol, but what about nitrogen? And this is burning process, so then nitrogen oxides there. Are they controlled somehow? How do you manage with this? Also, microorganisms which you use for your production, they are quite robust. They can very efficiently, like, ferment any of organic matter. So including the material of like animal, potentially human also, so very efficient. So that's why it must be control of their like production, their disposal, their they're just number, because we have already in our history of human, like, this development examples when we boost some biological species, even like animals, for example, cows. There's so many now, there are more than humans and wild animals, and they produce more air pollution than in Europe, at least, more than cars. So it's a very big problem now. So now we like use microorganisms. And then I think that we already, based on the previous experience, need to control this point. What can you say? What is done in this?
To the first question with the nitrogen, we have an anaerobic process, we have 75% of our nitrogen is ammonia, which is NH4 plus. And this is used as a fertilizer, or is used by the plants or taken up by the plants for their metabolism. The concentration is not worth to recover and to use it, for example, as an energy source. If I get your question right, it tends in the direction that we are harming the environment. Is it right, or did I misunderstand?
No, microorganisms, finally, they can go back to nature, because there are many, you just produce, and potentially, and this aspect must be controlled, because we have already also, again, another example, contamination of our environment with a vaccine-based, like, this microorganism viruses mainly. So it potentially can, and also heat from this microorganism, from your process. Do you collect this? Do you use this heat, or how?
So the heat is often used for heat supply of the industry, or for any drying processes. The one I mentioned from Stybush in Oregon. If you compare the let's call it contamination of organisms in your digestate compared to manure, then there is almost no difference. So the organisms are almost the same. We never found any contamination of microorganisms in your process. Even when you compare the contamination in black water from municipalities, here, if you treat it anaerobically, you have a degradation of the harming microorganism, or of the toxic, or however you want to call it, pathogens in or during the anaerobic fermentation. So it has more a positive effect on the pathogens than using it directly. So this is a self-regulating system, and the organisms, what you're spreading on the fields, are more the ones you are finding in the soils anyway. And by using it without any, for example, hygienization, while killing the microorganisms, then you are doing more a favor to the soils. And I hope it answered your question.
Okay, thank you. Let's believe that finally we will have really like
no, it's a long discussion.
Yeah.
We have I discussed many times with different people about it. It's always pros and cons, but there are many publications available where the degradation of the contaminants or pathogens took place.
Thank you.
Welcome.
Please answer questions.
May I?
Sure.
Yeah. Günther, can you tell us about equipment, maybe some specific equipment that you have in your university that you use to measure content of biogas or substrate and effluent? Maybe some specific or general?
Yeah. First of all, nice to meet you. I hope you're good. Yeah, so what we are using a gas analyzer or it's a infrared analyzer. We are measuring methane, CO2, H2S, and oxygen. And we are back calculating the nitrogen because the nitrogen is barely measurable. So we are not cheating, but yeah, if we have a discrepancy of some percent, then we take the oxygen concentration. Dry matter is an oven, 105 degrees Celsius. This is a standard, but this doesn't really work pretty well for 1 hour. Just keep it longer in the oven, then the data is more mature than with if you do it by the standard. For the rest, yeah, volatile fatty acids, it's HPLC as a standard we use. What is easier, but which needs also more knowledge, is using the FOSS-TAC titration value. So you're titrating with phosphoric acid. Don't ask me about the concentration. I think it's 0.1 molar. And then you have, you're titrating to pH 6 and then 4.4. Then you have the values, and then you know the concentrations. This is something what I did when I was abroad in Brazil or Argentina or Peru. This is the way to go, because HPLC is far too expensive. It's a bit more precise, but also far more expensive. And the other one is just a titration, and it gives you a hint about the stability in your process. Nitrogen concentration is measured by, you can do it via the cuvettes, or which is not very precise because you have the dark color in your digestate. This always gives you a very vague value. The other one is a titration in a Kieldal system. Hopefully I didn't forget anything.
Yes.
CO2 can be measured via the addition of sodium hydroxide. But you are just solving it, and then you see the volume level different.
Yes, thank you. So some of those methods we also used.
Please, more questions?
We are done. I guess we are done for today.
Oh, sorry. Yeah, you're sitting there since the morning.
Yeah, yeah. If I wish, maybe just a brief question, a little bit maybe complicated. I remember in one of communication, the European Union just in 2022, yeah, indicated that over the EU, just about 50% of via gas facilities, yeah, actually are working. If it's true, for your opinion, it's more operational, technological issue, or just economical, or what?
I can say the numbers from Austria. This is what I know by heart. They were built as for a renewable or for crops. So the calculation, the economics were based on crop digestion with a low crop price. And many of the plants are economically not viable because the cost, the price of the crops increased. Mm-hmm. Same in Germany, to the best of my knowledge. And yeah, there is an economic reason, but the other one is also the plant operator needs some support because it's a complete new technology. If you are a farmer, it's very hard to get used to a new technology, and there is always space for some optimization. This comes also into play and into these numbers. And then on top, yeah, broken machines, new investments is always increasing the capex, and then the efficiency or the economical viability of the plant is not given. Yeah, these are the three main reasons. Some plants are struggling right now in Austria because the availability of cheap residues is not given. So they are transporting the residues for long distance, and thus the gate fee increases of the systems. Yeah, but 50%. So in Austria, it's a bit higher, to the best of my knowledge. Yeah, but they can be also always more efficient.
Okay, thank you. So we have some challenges, yeah?
Yeah, no, the challenge is given. But the beauty of the industrial application is you know the amount of residues which occur during your process, and you can thus plan your plant in detail, and then you are responsible for your own residues. So this is maybe, these plants are, I think, in the upper range of the efficiency.
Okay, thank you. Thank you.
Are you sure that there are no more questions?
Yeah, sure.
Then thank you once again.
Thank you.
Okay,
And
yeah.
I wish you a nice evening.
Yeah, thank you. Thank you.
And all the best.
Yeah,
Ciao.
thank you.
Yeah.
Well, Anatoly, your final conclusion as the moderator.
Yeah, I just wanted to thank all presenters and all participants, and I find that it was a very interesting and educative session. I hope everyone shares this feeling and impression. And I'd like to pass the floor to Professor Yakimenko now.
Yeah, I only want to say from all of us again, thank you to not only our presenters and all of us and our moderator, Dr. Anatoly Girish. It's really professional moderation. And guys, we are done for today. Thanks all of you, all of us. And we are meeting tomorrow at 9:30 Kyiv time. Again, we have a lot of interesting reports. Environmental issue, economy, social section will be tomorrow, and public health. So I hope you really share this Anatoly assessment, and tomorrow we will have more interesting reports and more interesting information. Okay, thank you. It's all for today. Goodbye.